Hello friends, today we will learn about enzymology. This is the second video of enzymology. So let's begin. First, we have discussed about enzyme inhibition. In that, we discussed about reversible and irreversible inhibition. So I have finished reversible inhibition in first video. If you did not heard, then heard the um, first video of enzymology. This is the second video. In this video we'll start from irreversible inhibition. So the inhibitors bind covalently with the enzymes and inactivate them, which is reversible. These inhibitors are usually toxic substances which may be present naturally or man-made. Lodo, iodoacetate is an irreversible inhibitor of the enzymes like papain and glyceraldehyde 3-phosphate dehydrogenase. Iodoacetate combines with sulfhydryl groups at the active site of the enzymes and makes them inactive. Diisopropyl fluorophosphate, in short DFP, is a nerve gas developed by the Germans during Second World War. DFP, that is diisopropyl fluorophosphate, irreversibly binds with enzymes containing serine at the active site. Example, serine proteases, acetylcholine esterase, and let's know about suicide inhibition. In this type of irreversible inhibition, the original inhibitor is converted to a more potent form by the same enzyme that ought to be inhibited. Example, allopurinol, an inhibitor of xanthine oxidase, gets converted to alloxanthin, a more effective inhibitor of the enzyme. Now let's know about enzyme specificity. Enzymes are highly specific in their action when compared with chemical catalysts. The occurrence of thousands of enzymes in the biological system might be due to the specific nature of enzymes. Three types of enzyme specificity are well recognized: stereospecificity, reaction specificity, and substrate specificity. So, specificity is a characteristic property of the active site. Now let's know about stereospecificity of or optical specificity. Stereoisomers are the compounds which have the same molecular formula but differ in their structural configuration. The enzymes act only on one isomer and therefore exhibit stereoisomerism. Example, L-amino acid oxidase and D-amino acid oxidase act on L and D amino acids respectively. Hexokinase act on dehexoses, glucokinases on D-glucose, amylase acts on alpha glucosidic linkages, cellulase cleaves beta glycosidic bonds. Stereospecificity is explained by considering the three distinct regions of substrate molecule specifically binding with the three complementary regions on the surface of the enzyme. The class of enzymes belonging to isomerases do not exhibit stereospecificity since they are specialized in the interconversion of isomers. Now let's know about the reaction specificity. The same substrate can undergo different type of reactions, each catalyzed by a separate enzyme, and this is referred to as reaction specificity. An amino acid can undergo transamination, oxidative or deamination, decarboxylation, racemization, etc. The enzymes, however, are different for each of these reactions. Now, let's know about substrate specificity. The substrate specificity varies from enzymes to enzyme. It may be either absolute, relative, or broad. Now, let's know about absolute substrate specificity. Certain enzymes act only on one substrate. Example, glucokinase acts on glucose to give glucose 6-phosphate. Urease cleaves urea to ammonia and carbon dioxide. Now let's know about relative substrate specificity. Some enzymes act on structurally related substances. This in turn may be dependent on the specific group or a or a bond present. The action of trypsin and chymotrypsin is a good example for group specificity. Trypsin, arginine and lysine. Trypsin hydrolyzes peptide linkage involving arginine or lysine. Chymotrypsin cleaves peptide bonds attached to aromatic amino acids, phenylalanine, tyrosine, and tryptophan. Examples of bond specificity: glucosidases acting on glucosidic bonds of carbohydrates, lipases cleaving ester bonds of lipids, etc. Next point is third point, broad specificity. Some enzymes act on closely related substrates which is commonly known as broad substrate specificity. Example, hexokinases acts on glucose, fructose, mannose and glucosamine and not on galactose. Not on galactose. Now let's know about the coenzymes. What do we mean by coenzymes? The non-protein, organic, low molecular weight and dialysable substance associated with enzyme function is known as coenzyme. The functional enzyme is referred to as holoenzyme, which is made up of protein part, that means apoenzymes, and a non-protein part, that is coenzyme. The term activator represents the inorganic cofactor like Ca2+, Mg2+, Mn2+, etc. necessary to enhance enzyme activity. Now let's know about the coenzymes are second substrates. Coenzymes are second substrates. Coenzymes are often regarded as the second substrates or co-substrates since they have affinity with the enzyme comparable with that of substrates. Coenzymes undergo alterations during the enzymatic reaction, which are later regenerated. This is in contrast to the substrate which is converted to the product. Coenzymes participate in various reactions involving transfer of atoms or groups like hydrogen aldehyde, keto, amino, acyl, methyl carbon dioxide, etc. Coenzymes play a decisive role in enzyme function. Now let's know about coenzymes from B complex vitamins. Most of the coenzymes are the derivatives of water soluble B complex vitamins. In fact, the biochemical functions of B complex vitamins are exerted through their respective coenzymes. Now let's know about non-vitamin coenzymes. Not all coenzymes are vitamin derivatives. There are some other organic substances which have no relation with vitamins but function as coenzymes. They may be considered as non-vitamin coenzymes, example ATP, CDP, UTP, etc. Now let's know about nucleotide coenzymes. Some of the coenzymes possess nitrogenous base, sugar and phosphate. Such coenzymes are therefore regarded as nucleotides. Example, NAD plus, NADP plus, FMN, FAD, coenzyme A, UDPG, etc. Now let's know about coenzymes do not decide enzyme specificity. A particular coenzyme may participate in catalytic reactions along with their enzymes. For instance, NAD plus act as a coenzyme for lactate dehydrogenase and alcohol dehydrogenase. In both the enzymatic reactions, NAD plus is involved in hydrogen transfer. The specificity of of the enzyme is mostly dependent on the apoenzyme and not on the coenzyme. Now let's know about mechanism of enzyme action. Catalysis is the prime function of enzymes. The nature of catalysis taking place in the biological system is similar to that of non-biological catalysis. For any chemical reaction to occur, the reactants have to be in an activated state or a transition state. Now let's know about enzymes lower activation energy. The energy required by the reactants to undergo the reaction is known as activation energy. The reactants when heated attain the activation energy. The catalysis the catalyst or the enzyme in the biological system reduces the activation energy, and this causes the reaction to proceed at a lower temperature. Enzymes do not alter the equilibrium constants that only enhance the velocity of the reaction. The role of a catalyst or an enzyme is comparable with a tunnel made in a mountain to reduce the barrier, and the enzyme lowers energy barrier of reactants, thereby making the reaction go faster. The enzymes reduce the activation energy of the reactants in such a way that all the biological systems occur at the body temperature below 40 degrees centigrade. Now let's know about the enzyme substrate complex formation. The prime requisite for enzyme catalysis is that the substrate must combine with the enzyme at the active site to form enzyme substrate complex, which ultimately results in the product formation, that is enzyme plus substrate gives rise to enzyme substrate complex gives rise to enzyme plus product. Now, let's know about lock and key model or Fischer's template theory. According to this model, the structure or conformation of the enzyme is rigid. The substrate fits to the binding site just as a key fits to the proper lock or a hand into the proper glove. Thus, the active site of an enzyme is a rigid and preset temperature where only a specific substrate can bind. This model does not give any scope for the flexible nature of enzymes. Hence, the model totally fails to explain many facts of enzymatic reactions, the most important being the effect of allosteric modulators. Now, let's know about induced fit theory or Coastland's model. Coastland in 1958 proposed a more acceptable and realistic model for enzyme substrate complex formation. As per this model, the active site is not rigid and preset. The essential feature of the substrate binding site are present at the nascent active site. The interaction of the substrate with the enzyme reduces a fit or a conformation change in the enzyme, resulting in the formation of a strong substrate binding site. Further, due to induced fit, the appropriate amino acid of the enzyme are repositioned to form the active site and bring about the catalysis. The induced fit model has sufficient experimental evidence from the X-ray diffraction studies. Coastland's model also explains the action of allosteric modulators and competitive inhibition on enzymes. Now let's know about substrate strain theory. In this model, the substrate is strained due to the induced conformation change in the enzyme. It is also possible that when a substrate binds to the preformed active site, the enzyme induces a strain to the substrate. The strained substrate leads to the formation of product. The concept of substrate strain explains the role of enzymes in increasing the rate of reaction. In fact, a combination of the induced feed model with the substrate strain is considered to be operative in the enzymatic reaction. Now let's know about the mechanism of enzyme catalysis. The formation of an enzyme substrate complex is very crucial for the catalysis to occur. It is estimated that an enzyme catalyzed reaction proceeds 10 to the power 6 to 10 to the power 12 times faster than a non-catalyzed reaction. It is worthwhile to briefly understand the ways and means through which the catalytic process takes place leading to the product formation. The enhancement in the rate of the reaction is mainly due to four processes. First is acid base catalysis, second is substrate chain, third is covalent catalysis, fourth is entropic effects. Diagnostic importance of enzymes. Let's know about diagnostic importance of enzymes. Estimation of enzyme activities in biological fluids is of great clinical importance. Enzymes in the circulation are divided into two groups. First is plasma specific or plasma functional enzyme, and second is non plasma specific or plasma non functional enzymes. So let's know about both. First is plasma specific or plasma functional enzymes. Certain enzymes are normally present in the plasma and they have specific functions to perform. Generally, these enzymes activities are higher in plasma than in the tissues. They are mostly synthesized in the liver and enter the circulation. Example, lipoprotein lipase, plasmin, thrombin, cholinesterase and celluloplasmin, etc. Impairment in liver function or genetic disorders often leads to a fall in activities of plasma functional enzymes. Example, deficiency of ceruloplasmin in Wilson's disease. Second one, second one is non-plasma specific or plasma non-functional enzymes. These enzymes are either totally absent or present at a low concentration in plasma compared to their levels found in the tissues. The digestive enzymes of the gastrointestinal tract, example amylase, pepsin, trypsin, lipase, etc., present in the plasma are known as secretory enzymes. All the other plasma enzymes associated with metabolism of the cell are collectively referred to as constitutive enzymes. Example, lactate dehydrogenase, transaminases acid and alkaline phosphatases, creatine phosphokinase. Estimation of the activities of non-plasma specific enzymes is very important for the diagnosis or and prognosis of several diseases. Now let's know about the isoenzymes. The multiple forms of an enzyme catalyzing the same reaction are isoenzymes or isozymes. In short, isozymes we can say. They however differ in their physical and chemical properties, which include the structure, electrophoretic and immunological properties, Km and Vmax values, pH optimum, relative susceptibility to inhibitors and degree of denaturation. Example, lactate dehydrogenase, creatine kinase, lactate dehydrogenase, that is 5 isoenzymes, creatine kinase, that is 3 isoenzymes. Finally finished, I have finished the enzymology. Enzymology is a vast chapter. There are many topics on this. to study. In one two video, it's not possible to cover all the things. So overall, I have finished you can clear the concept of enzyme in my video. So any questions regarding enzymology, you can ask me in the comment box. I'll definitely answer the question. Thank you. For more updates, you can subscribe my channel. Thank you.